Oh, poor thing. All right, hi everybody. Just wait a minute or so for peeps to arrive, and then we'll get going. Yeah, hopefully any issues that people have had getting onto Zoom have been kind of ironed out. Obviously, if not, you're not going to be here to let me know, right? So it's kind of not very useful. Okay. All right, so I'm going to get going. 'Cause it's hit 9 o'clock. I want to make sure we finish on time. So first thing that I want to do today is something that I didn't get a chance to do on Wednesday, which is talk about expectations a little bit, right? And you're also more than welcome to ask questions at any point, obviously. So there's no two ways about it. And you've probably heard this already from other students that have taken this class with, me or Dr. Page or whoever else that teaches it. But this is a very difficult class. It's kind of for two reasons. One is because it's at the kind of the cusp between, or the pivot. Sorry, puppy's making a lot of noise. The pivot between kind of like the intro classes, which are, to kind of get you into the college way of doing things, and the more advanced upper division classes, which really rely on a lot of what you learn in this class to be able to teach you more advanced concepts and principles and look into a lot cooler things, right? So that kind of expectation is part of the reason why this is a difficult course. The other reason is that we've just got a lot of stuff to cover, and we're going to be doing it in greater depth than you have experienced before, right? So it's not just kind of the breadth that we have with Gen Bio 1, but also we're going to be diving into a lot greater depth, right? More detail, more mechanistic kind of stuff, essentially, right? How do these things happen? It's not just going to be a matter of, well, this does this and this does that. It's like, how does it do that? Why does it do that? What does that actually mean? Kind of like in a bigger sense, right? And that's really kind of where we want to take you, because it's important that you get to kind of really get a feel or a skill or ability to take information and kind of put it into different places, right? To kind of connect it to other principles and concepts you understand, start thinking laterally, right? Because we don't want to produce a bunch of automatons that can regurgitate facts, right? That's essentially Google, right? Or now, or used to be books or whatever, right? What we want are to educate students and to generate graduates, right? Because you're going to be going to do something with this afterwards, hopefully, kind of bigger and better things. But what we want to do is to produce graduates who are capable of that lateral thinking, that kind of, why is that doing that? Oh, well, I know something about that and something about this, and maybe those kind of work together. And kind of taking knowledge and understanding from different areas, right? And that's something that you can't get from just staring at a screen or a book, right? So that's why, kind of genetics is really quite a hard course, right? Because it's going to be a bit of uh, it's a step or two higher than Gen Bio 1 in terms of difficulty. And it's also just a constant onslaught, right, of information and principles and concepts and mechanisms and so on and so forth, right? So it's a course where, just like Gen Bio 1 in many ways, it's very easy to get overwhelmed and behind, right? So really a lot of what we do in this course is really kind of designed to keep you moving along, right? To kind of keep you up to date. But there's an awful lot of stuff that you can do too, which will make that easier, a little bit more uh, likely to be successful, I guess would be the best way of putting it. And the success rate, although it's better than GenBio 1, isn't super high either, right? So generally speaking, I would expect 55 to, I think maybe 65 is my highest percent of students get a C or better in this class, right? So out of, what's that, 35 of you, maybe 20 will get a C or better, and the remainder won't, right? And you need a C or better to progress in your major, right? So your next course most likely will be evolution, right? It might be micro, it kind of depends on what order you do things and so on and so forth. But evolution, at least, I don't know about micro, requires a C or better, right? And so you have to get that C, right? So there are a lot of different things that you can do to be successful, right? You don't have to be super smart, right? That's not really a requirement, right? But you do have to work super hard and you have to be very organized. And the key to that is one, doing all your homeworks and your lab stuff, right? Those two things are essentially 45% of your overall grade, like your course grade, right? So if you are super diligent about those, you get them all done, you get the highest marks you can for every part of that, you've already done a bunch of the heavy lifting, right? You're already, what's that? Most of the way toward, not most, kind of at least more than halfway towards getting a C, right? So don't forget that. Put any of those deadlines in your calendars, in your phones, or whatever, at least a day, notification, right? Before the due date, right? So you don't get caught up in other stuff and forget to do it or whatever, right? I'm as easygoing as I can be in terms of helping you. So if like crazy stuff happens or, for whatever reason you just forget to do a homework, just let me know and I'll extend that for you. But you still have to do the work, right? The other thing is essentially you're going to be constantly studying this class, right? Oh, hey, can you do me a favor and let Luna in? Thanks. Um, essentially you start today, right? And what I always advise students, and I know I'm not perfect at doing this myself, right? So I understand that it's hard, is set aside ideally 2 hours a day, right? Put it in your calendar, Monday to Friday, take the weekends off or save them for other things. But put in your calendar 2 hours a day. This is when I sit down. This is when I go through my course material. This is when I prepare for the class the following day, right? Um, so hopefully some of you have actually gone through the PowerPoint for today, so it's not going to be completely new. That's something I'd strongly recommend, right? And then, studying some of those facts, doing through like online quizzes, using the questions in the textbook to test your knowledge, study groups, whatever, right? That's got to be a constant thing, right? The reason why I know this stuff so well, like the material that we're going to be teaching, is because I've taught it a bunch of times. It's repetition, right? It's not because I'm a born genius at genetics. It's because I've worked hard, in my studies, and I work hard studying this material, and I've just taught it a bunch of times, right? So you're what is it, fifth semester I've been teaching genetics, fourth? I don't remember, something like that. And I've been teaching it since I started teaching nearly 7 years ago. So what you see is the cumulative result of that, right? It's not that this is kind of like you got to ascend through the clouds up Mount Olympus to the gods, and therefore then you will be a worthy one, right? It's just information and knowledge and understanding, right? So it's not beyond anybody, but it does take a lot of work, right? And so if you can get into that habit of doing that work religiously, and frequently, and asking questions in class and bringing those questions up, because you've got to figure out what you don't know before the exam. That's something that I kind of bang on about a lot. So most certainly you've heard me say that before at some point in some class, right? So you don't want to find out what you don't know in the exam, because by that point it's too late, right? You want to find out ahead of time. And that's what I'm here for. Your peers can help you with that. Even your family, right? You know, kind of like give them a textbook and say, quiz me and see how you do. You'll find out what you don't know pretty quickly, right? And that's a very valuable thing to do. I find out what I don't know frequently too. Actually, yeah, just the other day I had to look up something because I was like, what's that all about? You know, I don't know anything about this, right? So it's a constant process, right? Never stop learning, but it has to be a very organized, diligent process too. Now, if you do all of that, attend these Zoom sessions, ask questions, do all of your assignments and do them as best as you can, right? Getting the best grades that you can, then there's a pretty good chance, or better than even chance, to be honest, that you'll get a C or better, right? Even if you're not naturally academically gifted, right? Because obviously there's variations in how easy people find this stuff to learn, right? If you don't do typically even one of those things, then the chances of you passing this class with a C or better is much reduced, right? And my aim is not to exclude people from the biology major, right? My aim is to prepare you for the rest of it as best as I can, and you know, kind of say like wave goodbye as you head off to ever more interesting subjects, right? Than the one that I'm teaching you. Right, that's my kind of aim in life, really, with this course. Does anybody have any questions about any of that? Okay, another thing that is worth talking a little bit about, and we can talk about this again before the first exam as well. A lot of how I teach is based on my knowledge of what's important in a scientist, right? From my own experience. And typically what's important is the ability to think laterally, think outside the box, join different things together, understand principles and processes, things like that, right? General retention of knowledge is useful, um, because you do need to know stuff, but it's not the defining feature of a good scientist, right? And so uh, as a result, I teach a lot based on principles and concepts. If you listen to the kind of like the podcasts that I've uploaded, things like that, you'll see that I tend to focus on kind of key points which matter a lot. The facts are things which are good to learn, right? You can't really do this course without them, but they're not the key important point. And as such, in my exams, that's how I'm going to be assessing you too, right? There will be about, I don't know, 50, 60% maybe of basic recall questions, essentially things that you can look up in Google, right? And this is an open book exam, or these will be open book exams. I don't use lockdown browser or anything like that, but they are timed, right? So you don't have a lot of time to look up things. And also, even if you were able to find every single one of those 50 or 60% of the answers on Google, you still wouldn't pass the exam, right? So the rest of the exam is basically kind of how well do you understand the information, right? How well do you understand these principles that we're talking about, right? Why is an antiparallel DNA helix such an issue in DNA replication? What consequence does it have for gene structure, right? And chromosome structure, right? That's what I want you to understand. And so I don't know, probably about 2/3 of the rest of the questions are those kinds of questions. And the rest are really quite hard, not tricky, hard questions, which I will give you basically the beginning piece of information and the end piece of information, which will be, the answer, right? One of. And I expect you to get from that one place through potentially a series of steps to find the correct answer, right? And so you have to understand this material. Otherwise, though, that gap in between will be a chasm that you can't cross, right? And you're not going to find that answer in Google or a textbook or anything like that, right? You have to understand the material. And that's really where I kind of add value, as they say, right? So obviously ask any questions you like about the exam format and so on, probably a little bit closer to the time. First one's on 15th or 16th, something like that. But yeah, essentially that's kind of what you're going to be up against, right? And so they are difficult exams. They're not meant to trick you, but they are meant to assess your knowledge and your understanding, more importantly. Okay, so I'm going to get cracking and we'll start on where are we at? The actual material, right? So there's not a huge amount of stuff in the first PowerPoint that you really need to worry about. I think it's worth reading, particularly these. What's this slide saying? No, that's not that important. So slides 2 and 3 are good to understand, right? What actually is genetics, right? And you can boil it down to really the study of the hereditary material and how that material is inherited, right? Or traits are inherited. A trait is a kind of a catch-all word for a thing, right? Hair color, maybe. Phenotype being different colors of hair. And also, generally speaking, genetics is kind of split really between these three general fields, which really very much overlap. And really, we're talking about like the very, very small stuff, within a cell, which is molecular genetics, right? So it's what is DNA and how is it expressed and, turned into protein, things like that. How is that those traits transmitted or inherited, right, from one generation to the next? So this molecular genetics is really at the level of like individual cells, right? Transmission genetics is at the level of individuals, right? So me, my parents, my children, so on and so forth. Evolutionary genetics really scales that up to populations and time, right? So how do those traits change within populations over time, right? And really, there's a fourth one, which is a very new one, like really only in the last 20 years, which is genomics, right? And that's the study of a whole genome of an organism, right? And looking at the genetic makeup of both individuals and then comparing those between, species potentially to see how those things change over time. And the rest is some general history, which is worth knowing too. All right. So really what this PowerPoint is all about, and I think we get on to Mendel on Monday. Let me just double check. Oh, maybe. Oh, that's okay. I'm not going to stress too much about time. We've got extra time in there. So I have to check the schedule because teaching 2 classes, it's very easy to get confused what I'm teaching and when, particularly because they're different length lectures. So sometimes I'll get all kinds of confused about what exactly I've taught you and what I haven't. And so that's the reason why. Oh, Angelica, no worries about interrupting at all. In fact, if I don't hear you interrupt, just unmute your microphone, cough discreetly, and I'll stop. So note taking. So this is an interesting question, right? And it's very much depends on you as an individual, right? So what should we be doing in terms of taking notes, right? And so there are 2 things right here really that are critical. One, what are the key facts? Right? So and also what you can do is you can essentially almost pick up from what I spend more time on, right, are things that I think are more important than potentially other concepts or facts that you have in this PowerPoint. So what are the key facts, right? So for example, let's move this over here so you can see. All right, one of the key points of DNA is its directionality, right? So we can kind of do this as like almost a mock kind of note-taking exercise, right? So directionality, right? So DNA, RNA, and proteins have direction, right? So they are different at one end of the chain to the other end. They are, chemically different. And so that's denoted in this 5' and 3' kind of moniker, right? So now you're like, okay, he seems kind of taken with that idea about directionality. Let's make a note of that, right? And the direction in DNA and RNA is denoted by 5' and 3'. They're also called out here as key points in the slide as well, right? So if you didn't already know that, which you probably should have learned during Gen Bio 1, but might have fallen out since then. That's an important point to make, right? One of the reasons why it's so important is, I don't know what that was. One of the reasons why it's so important is that you can only extend a DNA or an RNA strand at the 3' end, right? You can't add new nucleotides, whether DNA or RNA nucleotides, at the 5' end, right? So the reason for that is this 3' hydroxyl group or OH group, right? That is what is essentially broken down in the dehydration condensation reaction, which you learn about in Gen Bio 1, to form these phosphodiester bonds, right? So that's kind of an example, right? So this one slide, the key points that you really got to get across is this directionality and how we call that out and what that directionality comes from, right? It's the kind of the phosphate group at the 5' carbon. It's the OH group on the 3' carbon, right? So if you get those points, that's great. I wouldn't bother making too many notes about that. Just kind of jot down the basics. If you don't, kind of draw a big circle around that, either on your PowerPoint or in your notes or whatever, and go, focus on this. Don't fully understand. Ask a question or look it up later or something like that, right? So your notes are really, and in many ways, your kind of guide to yourself your future self as to what you need to go back and study, right? So I think that's actually, but that's how I would work, right? So I can't tell you how best you will study because you're almost certainly different from how I am and how everybody else is. Everybody's different in some way when it comes to how you learn. But that's one suggestion, right? So kind of make it like a list of things to do almost, right? Oh, I know this, so I'm just going to put that in small. I really don't know this, or what the hell is he talking about? This is just gibberish, right? That'll have like double circle and then arrows and like asterisks or something around it, so that when you come back to like later today or maybe not today because it's Friday evening, maybe tomorrow when you want to go through your notes, those will be the ones that you need to do some more reading on, you need to find other sources of information, or those are what will be added to your list of questions that you're going to ask me on Monday. Right. So that's what I would suggest, right? So note taking not being a passive thing, right? I'm very big on active learning, right? Give me a second, Amber. I'm just nearly finished up. So use your note taking as an active mechanism for learning the material, not just to write it down and then because you've written it down, you don't need to worry about it anymore. But it's like a key to this is what we need to worry about. This, make sure you know it, but I'm pretty sure I already know it. So just like scan through it and then focus on these other things. Amber. Oh yeah, that's a good question. So for exams, the material from which the exams are pulled, or the questions in the exams, is almost exclusively the PowerPoints, right? So you will not be quizzed on stuff that is in the textbook that's not in the PowerPoints. Similarly, quite often, we'll, particularly when we get into like some of the cooler genetics, we'll go off on tangents, right? Those tangents will not be in the exam. They're just for your own kind of erudition, essentially, right? Sometimes, and typically the things I talk about most in the Zoom sessions will be the stuff in the PowerPoints that you really need to know, right? So, but on the flip side, simply staring at and studying the PowerPoints isn't necessarily going to get you the whole distance in terms of being successful in the exams, right? They contain concepts which you need to understand, and that's kind of where I come in during these Zoom sessions. So hopefully that helped as an answer. GenBio 1, yeah, so really what we're doing in a lot of genetics is we kind of step off from GenBio 1 and go deeper, right? So for example, DNA replication, right? We're going to go into that in more depth, right? More detail. But if you understand it from GenBio 1, you'll already have the foundation. And so that extra detail is not going to be too big of a lift, right? So a lot of what we do here is really just taking this a little bit further and deeper and then putting it into a broader context, which we couldn't do in GenBio 1 because we're essentially busy teaching you the foundation of that concept in the first place. So although I know it kind of depends. It depends on how well you got the stuff in GenBio 1, if it's worth kind of going back through some of those notes. It may give you a different perspective, right, which will help in understanding the material here. It's really kind of up to you, but it in some ways it does correlate, but it very much kind of, here's the GenBio 1 stuff, your foundation, like we're kind of starting there and then going up from that point. Cool. Good questions. All right, let's tuck that over there so I can see that if other people ask. So, right, anyway, so we've kind of gone over this slide, the importance of this directionality, right? And so really in terms of genetics, you've got to start with the very foundation of genetics, which is the structure of DNA. What is it made up of, right? What does it look like? And that has a bunch of consequences later on in terms of the material that we're going to be studying. So one of actually kind of, I go off in all kinds of tangents. To be honest, kind of give you a heads up, these Zoom sessions, I tend to bounce around a little bit, right? They're not necessarily completely linear processes because I'm kind of hoping, sort of assuming, but hoping that you would have had a look through them already before class, right? So this isn't just like me standing in front of you at the front of a class. This is more, well, this is cool and this is important. Oh, and this ties back into this, right? So I'm hoping to do a little bit more integration, I guess. Aaliyah, the direction is super important because for a variety of different reasons. Get to you in a moment, Helene. Direction is important because you can only extend DNA or RNA at the 3' end, right? So in terms of DNA replication, that means you have essentially, and let's kind of move on to this bit. There we go. DNA is an anti-parallel double helix, right? And so when you want to replicate that DNA, you're going to be replicating one in that direction, and you'd be replicating the other strand in that direction, right? And that causes all kinds of issues in DNA replication, which we'll kind of go through much later in the course. Similarly, if you have information going that way and information going that way, you can have genes going that way and genes going that way on opposite strands, right? So DNA isn't a single piece of information going in one direction because it has two strands in opposite directions. You actually have genes that can go that way and genes that can go that way, right? And if they overlap or they can share regulatory regions, right? So you can coordinately express two genes because they are pointing in different directions and have their regulatory region kind of shared between them. I actually discovered couple of those myself in my own work, even though I don't quite understand what that means yet. Right, so there's lots of reasons why direction is important. Quick question, Helene. Brush up on organic chemistry. Well, if you're studying OChem right now, that'd be a great idea. For this, the kind of chemistry that we get into is fairly straightforward, right? In terms of it's maybe later we'll talk about proteins a little bit. We'll talk about some of the different bonds. The main bit of OChem that you need to understand is that the bases are held together with hydrogen bonds, right? Here we go. Here's a question. If you can answer this, Helen, or anyone else can. Why are hydrogen bonds the perfect bond to hold together these two anti-parallel strands of DNA? Question to the class. Think about hydrogen bonds. Hydrogen bonds are what hold water molecules together until you heat them up, like boiling the kettle, and then you get steam. Are hydrogen bonds hard to break? That's all right. They are weak, right? They're weak individually, right? I mean, my favorite saying is like, oh, you put a steak in the kettle and you switch the kettle on, you're not going to have steak kind of steam flowing out the top of it after a little while. You'll just have like a really scummy kettle, right? If you put water in a kettle and you boil it, then those hydrogen bonds break, right? And those water molecules are then free to flow around. So hydrogen bonds are they're electrostatic bonds, right? So they're basically slight area of positivity here, slight area of negativity here, and they're kind of like those crappy magnets that only slightly hold things to the fridge. I don't know if you've ever, I don't know, I've got them all over my fridge. They're not like rare earth magnets, which you will kind of crush your finger if you get them between them. They're like the crappy kind of magnets that are on the back of like hooks and stuff, right? So individually they're pretty weak, right? In aggregate, they're reasonably strong, right? So that's why water boatmen can like walk on water and stuff like that, for example. So what do you need to do with DNA, right? So kind of I want to get the idea across here, right? Because this is a kind of foundational principle, but it really applies to a lot of different stuff. You know, when do we need to, sorry, I keep sticking my scuzzy finger up, but I've only got two index fingers. When do we need to break those two strands of DNA apart? Questions to the class. There's actually, generally speaking, two times we need to do that, two different purposes. Right, one of them's during DNA replication because you have to break it open. Yeah, Cameron, great question. Great answer, sorry. One is copying DNA, right? So you have to split it apart, access those bases inside, and then generate new DNA strands. Actually, that's actually another good answer, Jonathan. The other one is expression of DNA, right? So you have to get it apart so you can make an RNA transcript. DNA error proofing and repair. Sometimes that's actually an interesting kind of point. I haven't really thought of it like that. But yeah, for some of them, I mean, that's kind of essentially really small scale DNA replication occurring at sites of, errors and mutations. So yeah, I mean that applies too. Um, and so you have to hold these two strands together well enough, right, so that they don't just fall apart, right? DNA is a very robust molecule, and with isolated, sequenceable DNA from things like 60,000 years old, which is absolutely crazy, right? That's kind of that's like as long as Homo sapiens has been around for, basically, right? So that's kind of a crazy amount of time if you think about it. But also you need to be able to open up easily, right? So those hydrogen bonds are perfect for that, right? I would view them like as a zipper, right? You can unzip it, you can zip it back again, right? And it doesn't accidentally open up. Oh, so good question from Aaliyah about anti-parallel. Anti-parallel basically means that one strand goes in one direction, the other strand goes in the other direction, like so, like my fingers. Right. So it's just the same as if you're going down a country road, right? One lane goes one direction, the other lane goes the other direction, right? And so it doesn't really matter which way around you put them, right? So it could be like that, or it could be like that, right? It's exactly the same. Yeah, so it's based on the direction. One's going to be 5' to 3'. Yeah, I'll draw it up. So one is always going to be going like so. My finger isn't quite as well as I think it is. One is going to be going in the other direction. And because the direction is typically the direction in which they can be extended, we typically do the head of the arrow as 3' and the other end as 5'. So it looks like that. Oh, sorry, that's my daughter's telephone number. Don't want to have that up on there. Okay. Oh, I got a little bit optimistic about how quickly my finger had healed. So another thing that's really kind of important to get across, and again, is like part of this principle, is that these bases, the A, C's, G's, and T's, are hydrogen bonded, right? Together. Actually, technically speaking, it should be like that because it's anti-parallel, right? See, trying to be as accurate as I can. So does anybody remember probably from Gen Bio 1, how many hydrogen bonds stick those A's and T's and G's and C's together? And is it the same for A's and T's versus G's and C's? Pardon me. Question to the class. How many hydrogen bonds and is it the same? Jocelyn, I'm sorry, I don't know if I'm pronouncing that right. 2 is great answer. It's correct for one of those pairs. Can anybody remember which one? A's and T's or G's and C's? You're all going, oh, I remembered that once. Yeah, T and A and T's. Exactly. Right, so adenines and thymines are held together by 2 hydrogen bonds based on essentially the shape and the kind of the atoms that they have in their rings. Can anybody remember how many hold G's and C's together? It's not 2. Ooh, come on, someone have a guess. Well,
3.
guess the other way. Yeah.
Is it 3?
It is 3. Sorry, who was that? I couldn't see who was talking.
Raylene.
Oh, Ray, thank you. Yeah, it's 3. So the strength, right? Because each hydrogen bond has a certain kind of like strength, is going to be different holding together A and T's versus G's and C's. Now, kind of again, like kind of riffing off of that, if DNA has to be opened to replicate it or to transcribe it. At the sites where it gets opened, would you expect to find more A's and T's or more G's and C's? These are sites in the DNA, the double helix, where it needs to get opened, you know, repeatedly.
More A's and T's.
Yeah, exactly, right? So that inherent property of hydrogen bonds and how many hold A's and T's together is actually really important when you look at, gene structure or chromosome structure, because those are the points where you can open DNA more easily, right? Let's kind of look at it from, and I don't want to kind of like, bang on about this, but it's kind of cool to think about, right? Because this is a very like teeny tiny property, but it has very, very big consequences. Now, what about if you went down you were very, very lucky. I'm kind of very jealous of people who get to do this work. You go down in a deep sea submersible to a black smoker in the Pacific on one of the trenches, and you bring up some bacteria that live at, let's say, 105 degrees Celsius, right? Which is not the upper limit of temperature, but it's getting close. I think the upper limit's about 110 degrees Celsius, give or take, which is crazy, right? So what, in terms of their genome, would you expect to find more A's and T's or G's and C's? If that organism comes from somewhere that is like 105
T's
degrees
and
Celsius,
C's.
the other way, G's and C's, right? Because that will help that DNA of that organism stay stuck together until it needs to be pulled apart, right? Because they're in a very, very high energy environment, right? So something that's like a real kind of basic property of DNA actually has like real big consequences, even at kind of the kind of ecological levels, right? In terms of where organisms kind of live. It's like the selection pressures selected a higher GC ratio in, extreme hyperthermophiles as regards to, stuff you find on your skin or whatever, right? So that's just kind of a cool example about how you can look at this in like lots of different ways, right? And it's also one of the things that I like to think about too, because, I don't want to focus just on, genetics. It's also really cool to look at the bigger picture too, see how things fit in. Cool. Anybody got any questions? I've only done 2 slides. Okay. So I've got another 9 minutes or so, so we'll take our time. We don't have to rush. We've got Monday to finish going through this too. So anyway, so this is DNA is very much my analogy, which some of you have heard before, is that DNA is like a book in a library, right? So it contains all of the information, right? And essentially every single one of your cells has the same information in it, right? Although obviously all your cells are not the same as all the other cells, right? So, skin cells are very different from liver cells, which are very different from corneal cells, right? And even though they have exactly the same information or the same library of books, essentially, they are very different, right? And so the difference really comes from how those genes are expressed, right? And what those genes encode. And so one of another analogy that I have is DNA, the A, C's, and G's are like the letters in a book, right? The words are really these are called codons, right? So this is a codon conversion table. This is like a Rosetta Stone essentially for molecular biology. And so these codons are the words, right? You can think of genes as being paragraphs, chromosomes being chapters, so on and so forth, right? This kind of can stretch that analogy a little too far. But that information doesn't really do anything, just the same as a book in a library doesn't do anything, right? Now, if you want to bake a cake, which is a subject close to my heart, perhaps a little too close, you need to look up the recipe in the book, but that information isn't a cake, right? You actually have to do something and turn that information into something else, right? And so that's really what we mean when we talk about um, kind of gene expression, right? Turning, I can't see the top of that. Oh, there it is. Turning this information via a series of processes, which are pretty involved, and we're not going to go into these in great detail now, into something that does something. And typically, not always, but typically, that something would be a protein, right? There are also really cool examples of RNAs that do stuff too. Actually, here's another question. Can anybody think of an RNA that acts like an enzyme, right? It's not just an intermediate, it's actually a kind of biochemical actor. Can anybody think of an RNA that also acts as an enzyme called a ribozyme? And I'll give you a clue. It's on this slide. Technically, you got like 33.3% chance of getting it right if you just guessed. You're probably thinking, what? RNAs? Enzymes? No, those are proteins. Well, actually, RNAs can also be enzymes too. Very special types of one. All right, so I'll kind of narrow it down for you. Ribosomes. Yeah, that's a good answer, but not 100% correct, right? Because ribosomes are also made of protein, right? So the large and small subunit are made of protein. But what's the RNA that actually catalyzes the peptide bond formation? What RNA would you find in a ribosome? Ah, yeah, that's the one, Sammy. It's a ribosomal RNA or an rRNA. Pirates love rRNA, obviously. Um, so this is a RNA which does not encode a protein, right? Only mRNAs do, but it can actually do something too, right? So ribosomal RNA is the enzyme which catalyzes peptide bond formation between amino acids, right? But anyway, that's kind of a bit of a tangent. It's always just fun to kind of point that out. Most genes that encode things that do stuff, right? Most of them are protein encoding genes, which at some point will produce a functional protein, right? And so the process of going from DNA to protein overall is called gene expression, right? Or the sequence hypothesis, or, well, I mean, not quite right, the central dogma, right? And so that's made up of two different processes, which are quite different. One is we need to get this information out of the nucleus into the cytoplasm because translation occurs in the cytoplasm. And so this information here is going to get transcribed, right? Which is essentially kind of a fancy way of saying copied, right? It's not going to get copied exactly because we changed T's to U's and, you know, RNA is different from DNA, but it contains the same information, right? And that generates our messenger RNA. And then essentially these tRNAs act as uh, kind of the translators, right? T stands for transfer, but in another way of thinking about it is a translator, right? So each tRNA, let's find a picture of one. Here we go. Each tRNA holds only one amino acid and only one, right? Not just it can only carry one, but it can only carry one particular amino acid. And it also has a region on it called an anticodon, right? An anticodon is a series of three letters, right? Here's our word again. So this is like an anti-word, I guess. And it recognizes through the same kind of base pairing, right, between adenines and thymines and guanines and cytosines and DNA, it base pairs with the codon in the RNA, right? And so because this tRNA only has one anticodon, UAC in the case of this one, and it carries only one amino acid, methionine in this case. Wherever you have an AUG, which is a start codon at the beginning of the reading frame, you'll get a methionine in the amino acid sequence, right? And so really these things here, these tRNAs, are what is going to allow us to translate the messenger information in the messenger RNA into a amino acid sequence in the protein, right? So really, these tRNAs are like the Rosetta Stone, right? They allowed archaeologists to translate hieroglyphics into stuff that we could understand, sort of. Sumerian, I think, was the language that those hieroglyphics were translated into for fun knowledge, right? So we're gonna basically we're gonna finish off this. Just a few other bits and pieces to talk about, and talk a little bit about mutations, and we're going to do that on Monday, right? So not an awful lot of heavy lifting today. Hopefully, I got you thinking about some of this stuff, right? Because I very much want you to be involved in this process, right? A, because it's a lot more fun for me. I get a lot more out of, kind of working with students rather than just talking at them. And B, you'll also understand what we're learning a lot better if you're involved in that learning, right? So long story short, thank you for all the questions. It's awesome. Yeah, Amber, that's a great thing to do. And this is kind of, getting on my soapbox again. One of the hardest things about learning anything is figuring out what you don't know, because you don't know typically you don't know it, right? And so um, part of this is kind of, figuring out what you don't know, and then you can learn it, right? And once you learn it, you do know it, and then you find out something else you don't know, and you learn that, and so on and so forth. Yes, Angelica, I'll hang on after class and wait for other people to leave, and then we can cover any questions you have. That's pretty much it for me. I'll post this up on YouTube and the chat session too. And yeah, have a think about some of this stuff. Go through the rest of the PowerPoint before we meet on Monday. And if you have any questions, bring those with you. And in the meantime, have a lovely weekend. All right, I'm going to keep um, actually, yeah, I can keep this open. Just wait for everyone else to leave, and then I can chat with Angelica. Oh, you sleeping? Oh, it's pronounced as a J. Oh, so Jelani? Okay, I'm sorry. Never entirely sure the correct way of pronouncing people's names. Yeah, good question, Melissa. The sixth and seventh edition would be totally fine. I've put up the reading recommendations for both in the syllabus section on Blackboard. So, and by and large, they're pretty much the same. But the seventh edition has some slightly newer kind of information in it, but not really so much that it would make a big difference. So yeah, if you want to, good weekend to you too, really. You can save some money and get the sixth rather than getting the newer version. Yeah, do that. All right, Angelica. What did you miss on the beginning of the class on Wednesday? Uh, to be honest, I can't remember. Largely because I also taught another class. So I don't want to give you poor information. Most likely, we were just going over kind of course structure, going over like assignments, homeworks, where you'd find things, stuff like that. So if you, let's see if I can actually show you this. Let's wait for that to disappear. So is it this one? There we go. So all of these Zoom sessions will be uploaded into a channel playlist for this class. And so this one, and that was sent out, at least I think I sent it out yesterday, I think, because I was busy with lab on Wednesday. But yeah, here's me. Looks just the same, wearing a different t-shirt. So this will have the whole copy of what we talked about in the um, Zoom on Wednesday. So if you only missed 15 minutes, that's not an awful lot of time. Um, just go through that and see if there's anything important in there that you needed to know. It's probably the easiest thing to do. Okay, did that help? Cool. Okay. All right. If you have any questions about any of that, either email me or bring them to class on Monday. It's totally up to you. I don't mind either way. But yeah, if you have any questions, that stuff you didn't really get from that, then just ask and I'll answer them best as I can. Cool. Awesome. Thank you. Have a good weekend too.